Alan, did you want me to introduce Lydia?
Yeah, well, yeah, here we are. I guess we're ready to go. So welcome to another edition of Plant Talk that was started. It's been going on for probably over 30 years, since the late 80s, I believe, started by Dr. Suzanne Copter. So it's wonderful to continue it. So we have Lydia today, and Dr. Kara Rockwell will introduce her. So all yours, Kara.
Thank you. I'm glad I was finally able to get in. Apologies for that. I would like to take this moment to introduce Lydia Cooney. She is my former master's student. Lydia first showed up in my office in the fall of 2015, so it's been over 5 years now. It's amazing. And I had advertised for a master's student. I had an idea of what I wanted, and Lydia came in and she's like, well, that's wonderful. But she's like, here's what I'm thinking about. And so she was so passionate about her love for the pine rockland habitat. And she had a great idea about looking at the pine rockland hardwood hammock interface in the ecotone. And I was just so impressed with her passion and her obvious knowledge of the system that I couldn't say no. And I learned so much from her during that experience and really grew to love that ecosystem as well. And since that time, she finished her master's degree. She went on, I always call it her dream job with Fairchild Botanic Garden, which she had talked about endlessly working with the conservation team there with Jennifer Posley and Jimmy Lang. And I have since worked with her in the field since her graduation. And I am continually amazed by her knowledge. And I will tell you that if you ever work in the field, identifying plants, Lydia is somebody in South Florida. She is somebody you want at your side. She has an amazing knowledge of the plants, but she is also, her enthusiasm is just contagious. And she, despite all the mosquitoes, despite the heat, the humidity, if you're just having one of those days when you're ready to go back to the office, her enthusiasm for discovering plants is just amazing. And so on that note, Lydia, take it away, and I'm very proud of you.
Oh, that's a really, really, really nice introduction. Thank you so much, Dr. Rockwell. It's great to work with you. And it's really, really an honor and a privilege to be able to be at the seminar today, speaking with the biology department and all the students and faculty. So everyone, my name is Lydia Cooney, and I am currently a field biologist at Fairchild Tropical Botanic Garden. And today I'm going to be talking about the project that I was initially hired on, was to take on this project and work together with the team to do conservation actions for 5 South Florida endemic plants. And we call it the Fab 5 project. So that was such a nice introduction, and just a little bit more about myself quickly. I'm from Miami-Dade County. I was born and raised here in Hialeah. And just from my experiences growing up and with my family, just a little bit in my 3 to 5 square mile area, it was a little greener than the rest of Hialeah. So just through all of it, nature grew on me. And so when I went to the university, I started with biology. My interest was always biology, and graduated in 2014 after taking many plant classes at FIU. I took a lot of the botany electives that are offered, like local flora and economic botany, and taxonomy of tropical plants. And so that was my real focus. And I gained, when I graduated, I gained some field experience at the zoo and with the National Park Service inventory and monitoring. And later on, I started to work with Kara for my master's. I went back to FIU. And now some of my work interests, what I do is GPS mapping and monitoring the rare plants and the habitats in South Florida. I'm interested in managing and learning about the management of these habitats to support the plants in them, and also restoring these habitats. Through visiting all these sites, I've learned to appreciate the phenology and the different life cycles of these plants. So I'm growing more interested in that. I'm into horticulture, which is a nice fit with the Botanic Garden. And with all this monitoring that we do, we generate a lot of data. So I'm interested in some of the organization of that. And more and more, I'm growing interested in genetics. I think this is important for plants, learning how the population is structured. And just more on a personal note, I'm interested in history of South Florida, all the development that has come on here quickly. And I'm interested in plants, and especially tropical fruits. So also, before I go any further, I have to thank everyone that helped in this project to get it to be such a success. And I have to thank the conservation team, which is the other Fab Five. Here we are in this photo on the bottom. It's 5 of us plus Jennifer taking the picture. That's Jimmy, that's myself, Brian, the horticulturalist, and Sabine, the seed lab manager. And I have to thank all the volunteers that helped in many ways, the U.S. Fish and Wildlife staff as well, helped with seed collection and monitoring, as well as permissions. Talking about permissions, the Miami-Dade County Environmentally Endangered Lands, they're very, very supportive of the work that we do, and we work in close collaboration with them and the preserve managers. Also natural areas management, who work to restore those county lands that we did this work on. And also would like to thank the Zoo Miami conservation and research staff, because we also did some work with them, and they helped us access the pine rockland as well. So the Fab Five is really, the title is called Partnering to Safeguard South Florida's Most Vulnerable Plant Species Through Strategic In Situ and Ex Situ Conservation Measures. So as you saw in the acknowledgements, it was really, we needed to partner with all these organizations to get this work done. And these are the 5 species that we work towards those conservation measures. I would like to mention the work that we do in the conservation program at the garden is largely grant funded. And so this funding came from the U.S. Fish and Wildlife Service. These are federal funds that were administered by the state through the Florida Department of Agriculture and Consumer Services. So these again are the 5 species, and in the next slides, I'm gonna go into detail about each one. So first, the Florida bricklebush, Burkhelia moseri. It's a perennial herb, and it grows from a tuber that can survive the seasonal fires. And the plant does, from monitoring, we can see that it does senesce. You know, it's sometimes dormant. You can't find the plant in the dry season. And when it's flowering in the summer, these are those flowers, and the seeds are achenes like in the aster family, and they're wind dispersed. This is the seeds in maturity. And this plant is only found on the Miami Rock Ridge, so endemic to the Miami Rock Ridge in the pine rockland habitat. Next is the Carter's flax, Linum carteri, variety carteri. This is an annual herb, so it will live for just one season, or maybe 2. And so it's really dependent on producing a lot of seeds, a lot of fruits. And this is a magnificent individual that was very successful. And They have these lovely yellow flowers, and it's only found on the Miami Rock Ridge in a really narrow area in Miami. And you can get it confused with another pine rockland yellow flowered species. But if you look closely, you can tell them apart. It's in the details of the flower. So the next species, the sandflax, Linum erinicola, this is a perennial flax. So not an annual like the Carter's flax, a perennial. And its flowers are much smaller. They're just a quarter of an inch wide. And they also produce many fruits. The fruits are a capsule that hold up to 10 seeds. And these plants are very wiry, very thin. And this one, the sandflax, is found on the Miami Limestone Formation. So the Miami Rock Ridge is part of this limestone formation, but it also reaches into the lower Florida Keys. And so the sandflax also has a population in the lower Florida Keys on Big Pine Key and other keys. So like the other species, it grows in the pineland habitat, and it also likes the marl prairies, which are more seasonally flooded. So the next species, the Keys wedge spurge, Euphorbia deltoidea, subspecies serpillum, is in the spurge family Euphorbiaceae, and it's a perennial herb living for more than 1 year, and grows from a taproot. And it grows right on the rock and in really sunny, rocky crevices. And it's just a sprawling plant. It's only found on Big Pine Key. The next species, the Big Pine partridge pea, is a perennial herbaceous plant in the legume family Fabaceae. And the seeds, the fruits, are multi-seeded legumes. And this plant was historically known from more islands in the Keys, but through time, as fire suppression of the pineland increases the hardwoods, then this herbaceous plant cannot live. And also from some storm surge events, now this plant is really thought to be just found on Big Pine Key now, but it was found on other islands with the rockland habitat. So those were the 5 species of this project, and the objectives for the project were to collect seeds, genetically representative seeds, so seeds from all the populations, so that we can enter them into long-term cold storage, which is another word for seed banking. And some of these seeds also went on to be the material that we sowed for the translocations. So then that's objective number 2, the plant introductions and augmentations, which were identifying the places and assessing the sites where we would put the 5 plants in. And this involved a lot of planning, pre-planning for the experiments, then later the transporting and setting up where are we placing the plants, and having all those tools to dig into the rockland. So this is the results from objective number 1. We collected the genetically representative seeds from as many sites as we could visit for the populations, and we tracked carefully how many individuals we collected seeds from. And so here on the left is the results for how many seeds for each species we have at Fairchild. And I'd like to note that some of these are ex-situ generated seeds, so plants that we had in the collection that we used for seed bulking, so that we could really bank a lot of seeds And these are on the right stored at the National Laboratory for Genetic Resource Preservation. This is the national site for seed banking in Fort Collins, Colorado. So a requirement for this project was to send some seeds to there. And these are pictures on the bottom of all the seeds for each species, left to right, Burkhelia, the Linum carteri, Linum erinicola here in the middle, very small seeds. This is Kama Christa seeds, and also the Serpillum seeds, very small. So once we had access to these seeds, we generated a lot of information, new information about the biology and horticulture of these plants. We conducted germination trials and different experiments with the seeds. So part of the seed banking procedure is you desiccate the seeds. So you remove water from the seeds to be able to freeze them so that excess moisture doesn't burst the seed and kill the seed. And here we generated just very cool information about germination rates. And so on the left is Linum carteri germination, and on the right, Burkhelia germination. And the two colors represent populations. So the species seeds from two populations perform pretty similarly, very, very identically. They germinate at the similar rates. And from those seeds, we made those XC2 collections where we further, collected seeds. That's how we ended up with several thousand seeds of Burkhelia. It's from this XC2 collection, and they went into the seed bank. And also these plants went to the translocations. So the plants we started from seeds, they went to be the translocation plants. And these were the translocations. We did several actions. We did 14 or more, really, if you think about it. So we started in 2019 with all these efforts. And part of this planning for this was to maximize our efforts. So you can see that at one site, we did several different actions. So we tried to be efficient as possible. So with 3 different species at one site, we conducted several actions. And also with the Keys species, we knew that we had to not put it on the same island, but create a new population on another island this was important to decrease the extinction risk of these endangered plants. So you don't want to put it all in the same island, put it on another island. And with Burkhelia, I would like to know, it was a plant that we could really reproduce. And we were lucky that we could share some through the Connect to Protect network of Fairchild. So some homeowners in their Pine Rockland gardens, they also have some Burkhelia moseri plants. So with these translocations, it wasn't just about putting the plants, growing the plants and just putting them. We had some specific objectives with this work. We wanted to develop a better understanding of how to conduct this. So the outplanting techniques and the best practice for these taxas, how to best outplant. We also wanted to understand how these plants could be used in a restoration context. So we see that these restored areas, they have some ability to support rare plants. We wanted to test that. and also to understand the ecological needs of the taxa, these plants, establishing them from seeds and or from planting. And then a plus from these translocations, a positive note for all of this is that there was the restoration going on at all these sites. And we had a strong land manager support to do this work, and very an armada, a lot of volunteers to help us sow the seeds and transplant, water the plants afterwards, and even take pictures of us, pictures during in between monitorings. So this is some of the work that went into, for example, just one big day. This is just 3 species when they were going to Rockdale. So it involved germinating the seeds, transplanting, and this is in February, and then here in May, they're already to be planted. And these are the Linum arenicola plants that were planted and all loaded up, ready to go into the volunteer day. So Miami-Dade County had organized a large volunteer event, and we capitalized on all those extra hands to plant all these Burkhollia. So yes, so now I'm going to go into the results of all those actions. So with the Burkhollia seeds, we were interested to monitor how the seeds would germinate in the field. So in total, we introduced 24 seed plots, and each plot had 20 seeds. So we introduced 480 seeds across 2 areas. So we tested a rocky area and a sandy area. And we introduced these seeds in June 2019. And 2 days after we had already started to see germination, it was starting to rain and we didn't need to water the plots. A week later, we see that the peak was almost 50% for both sites. But in a week after, we experienced a drought, no rain, and the seeds did not survive that drought. Not as well in the sandy area. They held on a little bit longer in the rocky area. And this was in 2019 where we didn't stop monitoring, but there was a few left. This was just the seed introduction results. So we outplanted, there was Burkhollia, there was 57 in total. We planted 25 in the rocky area and 32 in this nice silver saw palmetto sandy area. And here in green is the survival. So in both areas, they survived very well, and especially well in the sandy area, but there were quite a little bit more. And in orange, we have the reproductive status. And with this, we just see that the Burkhollia are very strong flowers in the summer, and they decrease their flowering over the winter. They're not flowering at all. So this has been a really interesting introduction, and I'm looking forward to monitoring this in May 2021. And after 1 year, we did survey beneath the outplants, and we looked for seedlings. And we did find seedlings, and a lot more in the sandy area, quite a lot more. And so this is something we need to revisit this spring and check on this. So for the sand flags, here is our effort. In blue, we had 6 sand flax that we introduced to this area, and they survived for up to 3 months pretty well, about 50%. So 3 of them survived up to 3 months, and they were also reproductive, all of them. But just after 1 year, we just had one still flowering and producing fruit. And after 1 year in September last year, we also found, we recorded 2 recruits underneath an outplant. And so this needs more study. This is active. So we also tried, we especially tried seeds of the sand flax. In total, we introduced 900 seeds and spread in 2 different times, one sowing in June and one sowing in September. And something just very interesting to note is that we did not see germination until the winter. So regardless of the sowing season, we only begun to see germination in especially January. And in the field, they did not germinate above 20%. So only 20% of seeds emerged. And this is one that we tracked for quite a while. They're very tiny. But the seeds that did emerge, they survived, over 60% for the first 3 months. And the dry season very much affected them. But there are still some seedlings out there today. There's about 7 seedlings. So the Carter's flax is an annual, but we still tried outplanting it, and we did 18 outplants. At the peak, 3 months after, there was 7 alive, and 4 of those were reproductive. And so this annual was, a few of them were surviving and producing fruit, and they made 20 fruits in this area. This is where we outplanted them, up to 200 seeds they made. And the following year, we observed the recruitment of new plants. So after 1 year, we counted 12 new plants, and this is them in the landscape. You can see the different colored toothpicks that we use to mark the plants there in the landscape. And those last year, those 12 plants themselves produced 22 fruits, up to 220 seeds. And this in orange is that area where we found last year's recruits. And then very excitingly, we found 35 new plants this year. So last year's generation already, it's an annual, so it produced its seeds and they disappear. And now in brown here, these are the areas where we found a total of 35 new plants. So they're slowly but surely spreading, and this is very novel, and we're actively monitoring this. So those were the results of the outplanting of Carter's flax. And we also tried seeds. So we sowed the seeds in a contained area. We tracked the germination, and we were tracking germination close to weekly and then monthly. And this decrease in survival here is the same drought period that affected the Burkhellias. So in the field, they didn't germinate up to 20%, but there was emergence. So seeds were germinating almost continuously we were finding new germinants every month. And this data does not include when a seed was reproductive, but we were seeing some seedlings reach reproduction status as well. So now in the Keys, these are the results from our action in the Keys with the Big Pine Partridge Pea outplanting. So in total, we introduced 48 plants, and in the Keys, there are the key deer, which are a major herbivore of herbaceous vegetation in the Keys. And so we thought it would be necessary to cage and protect some of those plants from herbivory from the key deer. 18 plants in 3 cages. So 6 plants in 3 cages were protected from herbivory, and while 3 of them were not protected. And here in the graph, see that those that were not protected, so the dashed line, they did suffer. So there was a decreased survival rate amongst those that were not caged, while there was 100% survival in those that were caged after 1 year. And then also very interesting is that those that were caged, they had a strong fruiting rates. This is just seasonality, so they are not fruiting in the winter, but you can see that after the first year, those that were not caged were not fruiting much at all, under 10%, while those that were caged were over 90% of them were fruiting. And so they had a stronger reproductive output when they were protected from the deer. And we also introduced seeds and tracked the germination of different seed treatments. So in blue, we have frozen seeds. Red is fresh seeds. We also tried scarifying or nicking seeds and desiccated seeds. And frozen and fresh seeds, they emerge very, very similarly. And one thing that we can tell is that scarified seeds germinate very quickly, much quicker than the other species, but those that germinate are pretty much, that's it, because you are kind of, you risk damaging the seed when you nick the seed. And it seems that the scarified seeds survived the best, but we generated a lot of seedlings from these introduced seeds. They survived over 30%, those that did germinate. So there are many, many seedlings out there. This is after 1 year, they're busting out. They want to get out of the seed plot. And the last species, or one of the last species, the Keys wedge spurge, we introduced just seeds of this species. And in the field, they did not emerge over 30%. So not many seeds emerged, but those that did, they survived very well, over 80% until the dry season, which causes mortality in these plants, some plants. But very, very encouraging is that after 1 year, when we went to survey, we found 50 new plants in the landscape there on No Name Key. And this is us looking for seedlings because they were growing out of the plots, and they're very reproductive and making seedlings. So we also introduced the sandflax in the Keys, and this was just a seed introduction. And we introduced seeds in 2 areas. One we call the Spanish Channel, and one we call the Quarry Side. And it seems that fresh seeds, they were very similar in both sites in terms of emergence. And those that emerged did survive for 1 or 2 months. And also I'd like to note that we introduced these seeds in August of 2019, but just like at Rockdale, we did not see any germination until the winter months. So in January is when we saw that peak in emergence. That's when seeds started germinating. And survival was good until, you know, the dry season. Survival really went down, but there are some seedlings there, and we are going to visit next week in February 2021, and we will monitor the introduction again. So in summary for this project, we preserved these genetically representative seeds for these 5 species, endangered species in seed banks. And there was a total of over 22,500 seeds that we entered into seed banks. And from this work, we learned how to grow these plants, these pine rockland plants, and how to germinate their seeds at the best rates in the lab and potentially in the field. And through this, we tested the translocation methods. So how do we implement this for all the species? And from that, we created many new populations for these species in restored pine rockland areas. Yes, I would like to note that in the no-name key area, we could not do that work without the restoration of no-name key, which was largely hardwood. And that was done by IRC, the Institute for Regional Conservation. Their habitat management teams, you know, decreased the hardwood area on no-name key, and we could introduce those endangered key species into those restored pine rockland areas. And this is a very active project, even almost 2 years after the translocations. We are seeing the recruitment of seedlings for all the species, nearly at all the sites. And these pictures, these are all pictures from the translocations, either seedlings that germinated or an outplanting. And this one of the big pine partridge pea is a recruit. So that is a seedling that is already a recruited seedling that's already reaching a reproductive maturity. So this is very, very active and interesting. And with that, I would like to thank you all for your attention, and I open the floor for any questions. And thank you all.
Hi, this is Gene Rosenberg. I just have a general question about cold storage.
Oh, yes.
How long are seeds viable in cold storage? Does that differ by species? And does it differ in some systematic way, large seeds versus small seeds? I mean, I'm familiar, for example, with the Millennium Seed Storage Project up in Svalbard and, you know, in those up in the Arctic. And I'm just curious in general about cold storage of seeds for viable. How often do they have to be grown out to be viable?
Yeah, that's an excellent question. And I'll try my best to answer it, but it's definitely more in the arena of our seed lab manager, who is knowledgeable in the seed biology. but we are making an effort to test the seeds after they've been in the freezer yearly. Since the seeds were submitted to the National Laboratory for Genetic Resource Preservation in Colorado, I think they have their protocols. They have their own protocols to monitor seed viability. Some species have been studied already, like the big pine partridge pea or the Kema crista lineata. That has been studied by Kew for its viability in the seed bank. But for example, the deltoid spurge species, it was previously thought to be recalcitrant or incapable of storage in the freezer. But during this project, there was some seeds from 2016 that were in the freezer. And for this project, we took them out of the freezer and germinated them. And they germinated in a germination trial in the seed lab. They germinated 80% of those frozen seeds germinated. So it's promising, though they were first thought to be recalcitrant. So we're planning to monitor the viability now every year of our seeds. So it's upcoming, it's active. Thank you.
Thank you very much.
Hey, Lydia.
Hello.
I was curious for the logistics of outplanting. Was this like a lot of pickaxes in the rock and stuff like that?
For some of the sites, for sure. The Linum Carteri introduction site is pure rockland, but it is kind of gravelly. So it does have some give because it is a restored area. So the rockland has been disturbed or altered a bit. So it's rock, but it has some give, and so it's more gravelly. But since we were largely working in restored areas, it was a little easier to dig, to be honest. But we did have the pickaxes ready.
And were these timed well with the season? Like, were you trying to hit the rainy season just right, or was it challenging?
Oh, yes, we did try that. So it was the early rainy season. So it was also in an effort with the Miami-Dade County volunteer event. So they had a pre-scheduled volunteer event at Rockdale, and we capitalized that we had those volunteers ready. And so that was in May. So we did have to do some supplemental watering, but just until the rainy season kicked in for 1 month. It only took about 1 month. And the plants established after that.
How do you water those? I mean, is it just carrying a pitcher out there, or?
Yes, we had to carry a big, larger 5 gallons or larger of water, and we had it stash in these areas so that we can come back and water quickly. So we had a overcast there at the sites that we can just fill up smaller jugs and then individually water. So we had reserves of water out there. Yes.
Cool. Thank you. That's interesting.
Lydia.
Hello.
Hi. Which was it Linum arenicola that only germinated in January?
Yes, that's when we started to see germination. So the seeds were there waiting a couple of months, and then they started to germinate. And we weren't entirely sure if it was related to the photo period. And a researcher at UF, he was also working with the sandflax, Linum arenicola, and he has observed that temperature could be a factor in germination. So they could be responding to the cooler temperatures. So the linums, the flaxes are a little bit more temperate in nature, I do guess. So they're responding to that cooler temperature. Yeah.
Yeah, I mean, that was what I was wondering, why you thought that, how that would have evolved if that's a dormancy that's broken in the middle, in the middle of the dry season. There's got to be a reason for that.
Yeah, we're actively learning as we're doing this, and then that research into the seed germination in the lab too, from a UF researcher, brought that factor into play of temperature.
Okay, thank you.
Lydia, nice to see you again. It's amazing that you're doing this incredible work. This is just so, I mean, you're like a role model for our current students. This is just so great. But anyway, I'm wondering what you think the chances of any of these species being able to establish in the FIU Nature Preserve.
It's a potential thing that could happen. We can talk about it with the conservation program manager, Jennifer Pawsley, and there could be seeds to go around, and we can try a new novel introduction in a managed site like the FIU Pine Rockland Preserve. Yeah.
Be incredibly exciting just to walk out and see these rare and amazing plants growing right in our backyard.
Yep.
Right,
yep.
Thank you.
The Pine Rockland there at FIU is more and more being managed. And I think you guys had a fire last year too, in 2020. Is that right?
I know they did do one recently, but now I can't remember. I can't even remember what I did yesterday. So it's kind of hard.
Yeah, there's a fire at the nature preserve.
Yeah, I think the second one now in under 7 years. I think so.
All right.
Awesome.
Hey, Lydia, I just had a quick question.
Hello.
Start out with the these species, and the amounts that you're putting out there are relatively small compared to maybe what it was before they were extirpated from certain sites. Are you planting any sites where active burns are happening? The question about the preserve made me think about it, because obviously there are plants that are adapted. It's a fire adapted ecosystem, but is that a concern when you're dealing with smaller amounts of these rare plants, when you're reintroducing them? It's something you throw them out there and just see what happens, or is the idea to wait until they're actually starting to recruit, and then if the fire's patchy, it's obviously not going to wipe out some of those initial populations that you reintroduced?
Yes, so the results that I went into were pretty much site focused. So for one site, for example, we did introduce Burkholderia to 2 other sites besides Rockdale. We introduced it into Zoo Miami in a big restoration area, and also in Seminole Wayside, which is much more in a natural setting. So that site is very likely to see a burn, and if a burn does happen, the aluminum tags that we use to track the plants, I think they're pretty fire tolerant. So we would be able to monitor afterwards, and then it would be very cool to see if recruitment is stronger after a fire, or if the seedlings are just combusted. We haven't tracked recruitment at Seminole Wayside as closely as we have done at Rockdale, but there are seedlings there too. at Seminole Wayside. There has been recruitment there too. So there has been talk with the preserve managers, you know, if it does burn, you know, so be it. That's part of it, and they're very much in tune, the Burkholderia, to burn with the tuber that they resprout from. So, but I'm interested to know if they will burn the flax areas. I think that might not happen for quite a while. I think it's very low productivity there. It's very harsh. So there's not much grasses or anything growing there yet. There's just gravel. And so that doesn't really need a prescribed fire just yet. It's very open. Thank you for your question.
All right, anybody else? Well, thank you so much, Lydia. That was nice to have a positive story, seeing some natives, some endemics increasing around here. So that's nice.
Thank you all. It was really great to talk to you all about this work project that Fairchild is doing, and I get to organize it and talk to you all about it. Thanks for joining today.
Lydia, great presentation.
Bye.
All right, thank you all. I guess that concludes it. In 2 weeks, I believe we have our next one. So we'll see you then.